This is lecture 4.1 on ATP and enzymes. If you recall from the large biological molecules lecture, we discussed proteins as being a very common large biological molecule that have diverse functions in the body. One of those diverse functions includes function of enzymes. That's what we're going to focus on today. I'd like to remind you that a protein is made up out of amino acid monomers. In the picture on your right-hand side, you'll notice that the primary sequence of a protein is the sequence of amino acids. This sequence determines the sorts of folds and bends that result in secondary, tertiary, and quaternary structure. The secondary, tertiary, and quaternary structure determine the three-dimensional shape and thus the function of the protein. So if you change the primary structure by changing the sequence of amino acids, you could potentially change the quaternary structure or the ultimate function of the protein. Likewise, if you introduced factors that changed the secondary structures or tertiary structures, you could also change the three-dimensional structure of your protein and thus change its function. Secondary structures and tertiary structures get their stability from relationships between molecules within the primary sequence. So you may have things like bonds between amino acids where you have sulfur atoms. Those are called disulfide bonds. You might have hydrogen bonds between slightly positive and slightly negative sides of the molecule. And anything that can disrupt hydrogen bonds or disrupt disulfide bonds could in turn disrupt the three-dimensional structure. We'll get into why that is relevant in a few slides. What is an enzyme? It's a biological catalyst that promotes chemical reactions. A catalyst is something that reduces the activation energy and allows a chemical reaction to proceed. In your body, every single one of your chemical reactions is mediated by an enzyme, and that enzyme is specific to the chemical reaction. You'll be able to recognize an enzyme because its name ends with the suffix ase, ase. For example, lactase is the enzyme that breaks down lactose sugar. Helicase is an enzyme that breaks down the hydrogen bonds between two sides of a DNA molecule. An enzyme has a three-dimensional shape, and that three-dimensional shape allows a substrate, or the chemical that it will be acting on, to bond to the active site. They fit like a lock and a key into that active site. Once the substrate has bonded to the active site of the enzyme, they form an enzyme-substrate complex, and the enzyme-substrate complex flexes itself ever so slightly and changes shape. As it does so, it allows a break to occur between parts of the substrate, resulting in the production of new compounds from that substrate. The products leave the active site of the enzyme, and the enzyme can proceed to perform another instance of promoting another chemical reaction. Catalysts do not get used up in chemical reactions. They help to make them go faster, though. So an enzyme is not going to get destroyed. It will be reused over and over again within your body. I'm going to press pause on this. I added a link to a short video in here on enzymes and how they work. I suggest that you go into your Google Slides and watch it. It's really great. It'll help to summarize some of the ideas. So the function of an enzyme is to lower the activation energy and allow a chemical reaction to proceed. So without an enzyme, you might have a high activation energy, and with the enzyme, you have a lower activation energy. Each enzyme has an optimal function at particular temperature and pH ranges. So in some cases, what you would notice is that as the temperature increases, the activity of the enzyme increases until it reaches a certain temperature at which you get denaturation. So what is that? That's when your enzyme gets broken and it can't work anymore. The example that I like to give is that of eggs, because many of you have cooked a fried egg before. When you open an egg, the white of an egg is primarily a protein called albumin. And in the form that you get a raw egg, albumin results in a clear, liquid protein-water mixture. However, as you heat up that protein, you get denaturation between parts of the albumin protein. So you get the addition of heat, and that breaks and rebonds connections in secondary and tertiary structures. And you go from having a clear liquid egg to having a solid white egg. So the three-dimensional structure of the albumin has changed with the addition of heat. So most enzymes will increase their activity up until a point in which you get the denaturation. And the optimal rate is that peak rate. pH, or the amount of acidity in a solution, is also going to affect the ability of an enzyme to work. Acidity or pH is a measure of the number of hydrogen ions to hydroxide ions. Plain water has a pH of 7, and it has equivalent amounts of hydrogen ions and OH- or hydroxide ions. As you become more acidic, you have more H+ ions. And as you become more basic or alkaline, you have more OH ions. And those OH ions or hydrogen ions can interfere with the formation of hydrogen bonds between amino acids that result in secondary and tertiary structures. So if you add acid to a protein, you can actually change its three-dimensional structure. In fact, the example that I like to give is that of making ceviche. Ceviche is where you take a raw fish, like a white fish, and you add lemon juice or lime juice, and that's the cooking agent. And then, of course, you would add things like onion and cilantro for flavor. But it's not raw when you eat it, because the fish or the shrimp is actually cooked by the acid. The acid changes the three-dimensional shape. In fact, if you watch the fish or shrimp as you pour the acid onto it, it'll actually change from being translucent to being a whitish color. The texture will change from being very soft to being more flaky, like a cooked fish. So you can actually cook things chemically with acid. So your optimal function for each enzyme is going to be within a very narrow range of temperature and pH. And this is really obvious when you look at the major digestive enzymes in the body. Your body has to break down large biological molecules into smaller pieces so it can absorb them. Carbohydrates are digested by three primary enzymes: salivary amylase, which is in the mouth, pancreatic amylase, and maltase. Now, each of these has got a different pH level that it works at the best. Salivary amylase is in a neutral pH, whereas pancreatic amylase and maltase need a basic or alkaline pH level. On the other hand, protein is digested by enzymes like peptidases, trypsin, and pepsin. And these require different pH levels as well. Pepsin is released in the stomach and requires acidic pH level, whereas trypsin and peptidase require basic acidity. Now, if you look at the site of release of the major digestive enzymes, you'll notice a pattern. In the mouth, you have a neutral pH. As the food moves into your stomach actually releases a large amount of hydrochloric acid. It makes itself acidic, and that acidity allows pepsin to work. Now, if the food from your stomach maintained its acidity, you wouldn't be able to use trypsin or maltase to break down any of your other compounds. So as your food moves into your small intestine, your pancreas releases pancreatic juice, which is alkali or basic, and it changes that acidity, allowing those other enzymes to work. So one of your activity questions has got to do with drinking alkali water. So if you drink alkali water, it may be alkaline in your mouth, but once that water gets into your stomach, what's going to happen? Your stomach will simply produce sufficient quantities of acid to neutralize that alkali water. So by the time that water gets into your small intestine, it's not going to make any difference that you spent an extra $2 on that water. You might as well have just drunk tap water at that point, because your body has to regulate your acidity within very narrow ranges. Now let's talk about ATP. ATP stands for adenosine triphosphate. It's a complex organic chemical, and it's important for providing energy for living cells. It has three components to it. Adenine, which is the same adenine that you know from nucleic acids, ribose, which is a 5-carbon sugar, and 3 phosphate groups bonded to each other with high energy bonds. It is the energy in those bonds that is captured or released and transported. You can think of ATP almost as a rechargeable battery. When a phosphate group is broken off from ATP, it releases that energy and it forms ADP, or adenosine diphosphate. So we can use that energy for biosynthesis to make new molecules, detoxification of chemicals, thermogenesis, or the creation of heat, transporting ions, such as in active transport, or even contracting your muscles. Once the ATP has been decharged and you have your flat battery of ADP, you can recharge that ADP from a variety of different energy sources. Carbohydrates in particular are key to generating ATP. And in one of your later lectures, we'll talk about exactly how that happens. You can actually also generate energy from proteins and lipids. Lipids are particularly rich in energy because they have long fatty acid tails, and they're small packages of energy between the carbons in those fatty acid tails. ATP is generated by an enzyme called ATP synthase. And what is super cool is that ATP synthase is the same molecule in plants and in animals. And what it does is it uses a flow of hydrogen ions to drive a rotating component used to attach a phosphate to adenosine diphosphate. It's very funny to think about it, but it's similar to a hydroelectric dam. A hydroelectric dam is where you create a dam on a river, so you have a backup of water. And when you get extra water, what you can do is you can deliberately let small amounts of that water go over the dam and through turbines that rotate, capturing the kinetic energy of the movement of water. That kinetic energy can then be used to generate electricity. In a very similar way, hydrogen ions flow through the ATP synthase molecule, and it actually has that rotating component that is like a turbine, and that will actually help to connect phosphate to adenosine diphosphate to create ATP. In eukaryotes, ATP is made in the mitochondria, which have ATP synthase on their inner membranes. Slightly different in prokaryotes, but we'll focus on eukaryotes. The next video, I'm going to pause it right now. This next video that's embedded into your lecture is from Khan Academy. I recommend that you go back into your lecture slide and you watch it and take some notes on the details. It actually shows a really nice three-dimensional structure of ATP synthase and how it moves as it's generating ATP.